Hello and welcome everyone to this podcast for genetics. In this podcast, we're going to focus on Meiosis II. And then in the next podcast, we'll focus on how the products of Meiosis II are developed into gametes. As we begin this discussion of Meiosis II, let's take a brief step back and think about how we got to where we are, where we can talk about Meiosis II. Remember, we had initially two sets of chromosomes. We had this one homolog here and this one homolog here. So one homolog here, one homolog here. It then went through interphase, where it went through G1, S phase, G2, so during interphase, which was G1, S, and G2. And we generated this cell that then entered Meiosis I. The cell then went through Meiosis I, prophase 1, metaphase 1, anaphase 1, and telophase 1. And at the end of that, we generated two cells that looked like this. Okay, and remember, we had that crossing over event that allowed for the transfer between these homologous chromosomes. And that independent assortment during anaphase 1 allowed this blue chromosome to assort with this red chromosome. And same thing down here. Now what we're going to do is we're going to take these cells here. So this is at the end of Meiosis I, at the end of telophase 1. And now these cells need to enter Meiosis II. All right, so now let's have these start Meiosis II. And before we do that, though, I want to draw in our cohesin and sugarshin protein. So I forget which color I used on the last one, but remember our cohesin here are proteins positioned between the sister chromatids. So up here I'm going to write cohesin are the green proteins. And remember, there were cohesin proteins that were holding these homologous chromosomes, this homologous chromosome with this homologous chromosome during Meiosis I, but then the protein separase came along and degraded those cohesin proteins so that these two homologs could be separated. And the reason these cohesins weren't degraded, if you recall, was because they were surrounded by another protein called sugarshin. So I'm going to draw over here just so we can see what I tried to draw here, but it's a little limited in size. So the sugarshin and cohesin are here, with the cohesin in the middle, and it's protected from degradation by the sugarshin binding to it. So separase cannot degrade the cohesin protein. So I'm going to call this part down here, these cells here, prophase 1. Prophase 2. And this is mainly signified by the chromosomes condensing. I didn't mention it at the end of telophase 1, but during telophase 1, as the chromosomes are being pulled apart and you're making separate cells, the chromosomes themselves lose some of that structure and unravel from this condensed structure. But now in prophase 2, they reform that condensed chromosome appearance. What will happen now is the cells will enter into metaphase 1. And this is a pretty important thing that happens here. At this metaphase plate, just like we saw with meiosis, the chromosomes are going to line up. And they're going to line up because of these centrosomes. These centrosomes, which are going to form here in prophase 2, these centrosomes are going to attach to the chromosomes and position them along this metaphase plate. And let me draw them first, and then we'll talk about their importance here. So what I have drawn here, again, are cells that are in metaphase 2. And these spindles have positioned the chromosomes along this metaphase plate. But there's one big difference between what we see here and what we saw in Meiosis I. And that is, in Meiosis I, homologous chromosomes were on either side of this metaphase plate. In metaphase 2, what's on either side of the metaphase plate here are sister chromatids. So that's an important distinction I want to write down. Sister chromatids on either side of metaphase plate. And that's important because when we see anaphase 2 and telophase 2, that's when you're going to see them pulling apart. So they need to be along this metaphase plate. Now the sister chromatids are still held together because of cohesin and sugarshin holding them together here in between. So the hallmark of metaphase 2 is that they line up along the metaphase plate in such a way that on either side of the metaphase plate are sister chromatids. Next, I'm going to draw anaphase. Each cell goes through anaphase. So let's write anaphase 2 here, and let's draw the chromosomes. So as we see, as I've drawn here, the spindle poles from the centrosomes have attached to the chromosomes and have started pulling them apart. Remember here, they're still lined up along the metaphase plate. Anaphase 2, they begin to pull them towards one side of the cell or the other. And also down here, I seem to have forgotten to draw this part. Let's do that real quick. And the only reason this is allowed is because our good friend separase enters the picture again. And it can enter this picture again to degrade the cohesin because sugarshin begins to degrade at this time too. So those are two key points here to mention about anaphase 2. I'll write it here just because I don't have room over here to write it. Sugarshin degrades. When sugarshin degrades, separase can now do its job and it will begin to degrade cohesin. So cohesin also degrades because it's no longer protected by sugarshin. if there was a mutation that prevented sugar from degrading, you would not be able to pull these apart. We're at anaphase 2 now. Let's move down to telophase 2. And what I'm going to do is I'm going to erase everything here and start drawing back in this direction. Okay, so now let's talk about telophase. Each one of these now cells enters telophase, and what we're going to see is that we will now divide the cytoplasm for each of these to produce 2 cells from each of these, so for a total of 4 cells. What also happens is that the spindles are done, pulling it back. They pull all the way back to their proper place, and now each cell is going to have these 2 chromosomes in them. The nucleus also reforms around the chromosomes, just like so. And now what happens is that these can now go on to form gametes. At this stage here, they're not matured into gametes. They have to go through another process, which we'll talk about in the next podcast, to form specialized structures that we see in oocytes and we see in spermatozoa, or we see in spores if you're talking about fungal species. What I'd like to do, just real quick here, is to highlight a couple things. One, let's draw a line here. And over here, I'm going to draw the diploid cell we started with. So we can kind of look at it and see how it's different now. So this diploid cell initially had 2 copies of this larger chromosome. These were the homologs it started with. And 2 copies of this smaller chromosome. This homologous chromosome it started with. Through the process of meiosis 1 and meiosis 2, we ended up with these. Remember, meiosis 1 is what helps us understand how we got that crossing over that allowed some of the red chromosome to come over here and some of the blue chromosome to come over here. Anaphase 1 helps us understand how sometimes this blue one here could have got the small red one, or it could have got the small blue one. Anaphase 1 is responsible for that independent assortment. You can also see another big difference here because meiosis has 1 round of DNA copying, 1 S phase, but it divides the cytoplasm twice. So you end up with 4 cells. So 1 cell makes 4 cells. And in each of these cells, we've changed the ploidy. This is 2N, or diploid. Here, we have each of these cells are 1N, or haploid. So from here to the gametes, we can see the cells have changed in ploidy, and the chromosomal arrangements have changed a little as well. I'm going to erase this. I'm going to talk about one last thing. All right, what I'd like to do now before we close is I'm going to do a quick comparison of meiosis. I just want to make a few comments here. You should know that meiosis, crossing over, which all occur during meiosis 1. Whereas mitosis, the goal of mitosis, is to generate identical cells. Maybe actually I should write up here instead of diversity, I should say diverse cells. Here, let's say, remind you that in mitosis, you go from a 2N cell to a 2N cell, a diploid cell to a diploid cell. My cells, when they divide, they go from having 46 chromosomes to having 46 chromosomes, 23 pairs of chromosomes. In meiosis 1, you go from a diploid cell to a haploid cell. Remember, in meiosis 1, we pull apart homologous chromosomes, so that in each of those cells, there's just 1 copy of that homologous chromosome, so it's a haploid. The other homolog is in another cell, so we say it's a haploid cell. Meiosis 2 takes this 1 N cell, both of those 1 N cells, and makes 1 N cells. In this way, mitosis and meiosis 2 are more similar, because you go from 1 ploidy 2N to the same ploidy 2N. Meiosis 2 goes from the same ploidy, 1N in this case, haploid, to haploid cells. It accomplishes this because of this third point I want to make, and it's about metaphase. So, in mitosis, sister chromatids are separated. So if this was a cell in mitosis, you would have your metaphase plate here, you would have your copied chromosomes like such, and they would be separated along this metaphase plate so that each of these cells would get 1 copy of it. So it's a little out of order. I'm going to say meiosis 2 here. Meiosis 2 does the same thing. You have these 1N cells with 1 copy of every homolog, and it also lines up on the metaphase plate such that a sister chromatid is on either side. So this is another way that meiosis 2 is more similar to mitosis. In meiosis 1, homologous chromosomes are separated. So it might look something like this, where you have your metaphase plate here and a homologous chromosome on either side of that metaphase plate. The last thing I want to talk about in comparing mitosis with meiosis is that mitosis, you go from 1 cell to 2 cells. Meiosis, you go from 1 cell to 4 cells. You'll see on the next podcast that we can have different sizes of these 4, and sometimes only one of these will be functional, as we will see in oocyte development, but you will still produce 4 cells. All right, that's all I have to say about this podcast on meiosis 2. If you have any questions, please let me know. If not, I'll see you in class. Bye.